Hi, I'm Brett, lab tech and instructor here at PCC Cascade, and today we are going to be doing a virtual walkthrough of the Biology 101 Lab 4, Exercise 2, looking at cell size and surface to volume ratio and seeing how that affects the rate of material export. So to do this, we have some salty agar cubes that are going to stand in for, we could think of these as models of cells. And I've got 3 of them here. One of them we are going to leave intact. One of them we're going to chop into 8 pieces, and one of them we will chop into 27 pieces. So we have progressively smaller cubes that all have the same, or roughly the same volume to start with. So what we're going to do is I'm going to measure the cube that is going to stay intact. And this looks like it is 3 centimeters by 3 centimeters by 3 centimeters. I'm going to record that data. And then the next cube, I'm going to cut that in half in each of the different planes to give us 8 cubes. And I'll try and be careful when I cut that. Get it exactly in half, but it's not going to be perfect. Okay. Okay, so now we have 8 cubes. And I'm going to measure one of them and put the rest in this tray here. So this is about 1.5 centimeters by 1.5 centimeters by 1.5 centimeters. So I'm going to say 1.5 for all of those. And this last one I'm going to cut into thirds to make a bunch of really tiny cubes. For those playing at home, you can probably guess what the measurements are going to be, but we will verify that. Okay, so I'll just grab a representative cube, and that looks like 1 centimeter by 1 centimeter by 1 centimeter. Okay, I'll put all of those in that third tray. So how this experiment is going to work, we're going to put these cubes into deionized water, and then we're going to see how salty that water gets. So we have here some water that we have pre-measured to exactly 400 milliliters. And again, for those playing at home, you will need to figure out how many liters that is for some of the calculations we'll do later. Actually, while I'm here, I will put some deionized water in the other beakers. Again, we've measured exactly 400 milliliters of that. Okay. And just to prove to you that this water is not salty to start with, we have a Vernier LabQuest data logger connected to a salinity probe. And we'll put that in and we'll zero it in the first beaker is reading roughly 0. Might bounce around a little bit. And then we'll try this in the other beakers just to show that they're all the same level of not salty to start with. Yeah, they're all basically reading 0. Yeah, close enough. Close enough. So, so I've set the data logger to collect a sample every 5 seconds, and I'm going to start with the big intact cube that was 3 centimeters on a side. I'm going to put the cube in, and I'm going to stir the water for 2 minutes, and it'll be collecting data. But basically, we just need the starting point and the end point to see how much total salt came out of this cube. So we will start the data collection, put the cube in, and start stirring. This has a graph of the data. Actually, here I'll put it over on the meter mode so we can see how it's changing. Those are some unusual numbers. Well, the data might be a little bit noisy, but that's okay. Go over to the graph and see where we're at time-wise. Just got a little bit more left in this run. Okay, we are done, and the final reading, let's look at the data table. The final reading was 259 milligrams per liter. So I'll record that number. 59, and that was for the large cube. Let's save that. Oh. Okay. So I think I've saved that. We're going to repeat this experiment, but instead of the 1 big cube, we're going to use the 8 medium-sized cubes. I want to make sure I don't get any of the salt from the first beaker into the second one, so I'm going to rinse the probe off. I can double check that it still reads 0. All right, and here we go. These are kind of sticking. Oh no, there we go. All right, so now we have the small cubes, or not the small ones, the medium ones. Yeah, we'll maybe go over to the meter mode so you can Get a better look at the numbers. We're already approaching the saltiness of the one big cube. I think that was 259, so now we've gone past it. Gone way past it. Go back to the graph to see how much more time we have. Not too much more time to go. Again, the data's a little noisy, but the overall pattern you can see. And we are done now. Let's see what that last data point was. For the medium-sized cubes, at 120 seconds, we had a value of 833 milligrams per liter. We'll save that data again. Okay, and now we will once again rinse off the probe. Show you that it starts at 0. All right. Yep, basically 0 to start. We'll get all our 27 tiny cubes ready, and we will start that. It goes stirring. Again, I'll switch over the meter so you can see how that's going. And for the medium cubes, our value was 833. Let's see if the small cubes exceed that. Feel free to hypothesize while you're waiting. Oh, and we're at 908, so. The data's still a little bit noisy, but the overall pattern should be as expected, or be pretty clear. So again, we'll go over to the graph. And it looks like we are now done. I'll read that final data point off. And that is 1,482 milligrams per liter. So there we have it. We have the data collection, and you will have some numbers to crunch and see how this experiment turned out.